Hello everyone, welcome to today's live broadcast where you can ask the expert, Mammalian Transient Expression. I'm Rich Altman of Amgen and I'll be your moderator for today's event. We are delighted to bring you this opportunity to chat with our expert. This opportunity is sponsored by Gibco, part of Thermo Fisher Scientific. For more information, please visit www.thermofisher.com/protein expression. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. Our expert will try to answer as many questions as he can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this session, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. I would now like to introduce our expert of mammalian transient expression, Dr. Jonathan Zmuda. Jon is a director of cell biology in the Life Sciences Solution Group at Thermo Fisher Scientific in Frederick, Maryland. Jon leads a team of scientists dedicated to discovering and developing new technologies and products useful for cell biology applications, including transient protein expression, advanced cell culture, and rare cell analysis. Dr. Zmuda received his PhD in cell biology from the University of Maryland, College Park, and his undergraduate degree from Dickinson College in Carlisle, Pennsylvania. I'd now like to turn it over to Jon for the Q&A session.
Great. Thank you very much, Rich, for the kind introduction, and welcome back to all of the attendees that have been at the earlier sessions today. This session, as Rich mentioned, is called Ask the Expert. However, as evidenced by all of the fantastic presentations that we've had already today, you are all the experts out there globally, and I will humbly do my best to address any questions that you would like to ask of me. So at this time, you are free to please type in any questions that you have related to the topic of mammalian protein expression today. And we've got a list of questions that have already come in as well. That we can begin with as the questions continue to roll in. But I do encourage you to please ask any questions that you may have. So I will start with one of the questions that came in today, and it's somewhat of a global question, and it's, how do you see two mammalian systems like XB293 and XB-CHO fitting together in terms of a harmonized and efficient transient protein expression workflow within biopharma or biotechnology? And I think this is a really interesting question, and it has lots of different answers, and different companies will find different ways that high-expressing transient systems fit into their particular needs and workflows. As many of you know, XB293 launched about three plus years ago, and has had a tremendous response from its users. And it really was the first time that we've been able to attain these gram per liter levels of protein for some proteins in a transient system. And users very quickly became very comfortable with the XB293 system. They tailored it to their needs, and they used it often as the workhorse expression system for their transient protein work. At the same time, 293 cells, and XB293 in particular, was sort of filling a gap where transient CHO didn't really exist, or existed in a manner that could not provide the levels of protein necessary to do the work that researchers wanted to with CHO-derived materials. So oftentimes, even in the case of clinical candidates, drug discovery, early work was being done with 293 cells. And then at some point, when a decision was made that this candidate therapeutic would move forward in the development process, a switch was made to generation of stable CHO material. And certainly these days, with the advent of many new types of second-generation monoclonal antibodies that are very dependent upon differences in post-translational modifications, specifically glycosylation in the manner of afucosylated antibodies, et cetera, that can dramatically impact the efficacy and potency of these antibodies, there was a significant risk in moving from in vitro models and potentially small animal models in which the protein was expressed in the 293 system to bridging that over to toxicology and clinical studies where the material is now made in CHO cells. So it's very well known that there's quite significant differences in post-translational modifications between 293 and CHO. And again, certainly in these newer generation molecules that are highly dependent upon these, it's less than desirable to have to switch midway in your drug discovery process from one cell type to another. So when we set out to develop the XB-CHO system, our main goal was to allow for researchers to have the CHO-derived protein that they need at the earliest stages of the drug discovery process and transition that all the way through to stable cell line generation and eventually clinical trials and approved biologics. And so that, with the advent of the XB-CHO system, there's now two high-expressing mammalian transient systems that live side by side, and there's some question in terms of how to best utilize both of them in terms of everyday workflows. Now, certainly 293 has its niche, in my opinion, with regard to the generation of protein reagents that might be used either for immunizations or as therapeutic targets, whereby the human post-translational modifications would be highly desirable in these types of proteins. Whereas CHO certainly makes the most sense in terms of target drug candidates in which they would eventually be manufactured in stable CHO. But I think the nice interplay between the two systems also comes in the ability to have two systems with the potential to generate the protein that you need, even if it's a hard-to-express protein, at the levels that you need. So if your workhorse is XB293 and you find that certain proteins don't express well, or you've had instances where if that doesn't work for you, the next question is, where do we go and how arduous is the path going to be to get that protein up to and including, spending 4 to 6 months making stable cell lines. Certainly XB-CHO now provides an alternative system in which some of these hard-to-express proteins in 293 can be made. And in our experience and in many of our discussions with users of the systems, it seems as though there are certainly distinct differences in how the two systems work. You heard a great presentation from Puneet at Teva Pharmaceuticals earlier today on the differences and similarities of protein between XB293 and XB-CHO. And certainly, living side by side with one another, they should complement each other. And it may be that some end users find specific paradigms for where 293 fits in and where CHO fits in. In other instances, it may be more fluid. But I think at the end of the day, again, the goal was with XB-CHO was to streamline that drug discovery process so that work can start in CHO and stay in CHO. And then certainly for each of the systems, weighing their pros and cons in terms of where they best fit into your workflows, in terms of scalability, screening, post-translational modifications, hard-to-express proteins, etc. So while there's no single answer to this question, and I think it's something that will be user-defined in the end, we now have two tools in our arsenal that allows us the best shot possible to have the amount of transient protein that we need to do our work. So I'm hopeful that at least gave some insight on thought process between how multiple high-expressing mammalian systems can live side by side with one another in a protein expression workflow. Okay, great. So I have some other questions that have come in that are a bit more specific. Oh, actually, another question just came in. So I had a question that just came in on whether or not we see similar post-translational modifications between transient and stable CHO. And so that's a great question, because at the end of the day, if the post-translational modifications of a transient CHO system were quite distinct from what is seen in stable CHO, that somewhat limits the utility of the comparability of material from discovery through stable cell line generation. And so what we have looked at to date, and by no means has this been exhaustive, and this work continues on as we speak to have a much more succinct and clear understanding of post-translational modifications within transient CHO and between transient CHO and stable CHO. So to date, the vast majority of the comparability assessments in PTMs has looked at glycosylation between CHO-S cells, which is the parental cell line for the XP-CHO-S cells, in a stable CHO-S system versus a transient XP-CHO system. And what we have found here, and is called out in my presentation that you'll be able to download following the conclusion of the virtual event, we've seen that the glycosylation patterns of transient XP-CHO are highly similar to those of stable CHO-S. And this manifests itself, in the case of one molecule at least, a human antibody, in the predominant glycoform being G0F for both stable CHO and XP-CHO, with lesser amounts of G1F glycoforms. And if you contrast that with XP293, XP293 for the same molecule showed about 50% G0F, and then a much higher percentage of G1F glycoforms. So to date, and with our relatively limited comparison studies between transient and stable CHO, we've seen that the glycoforms seem to be quite similar between transient and stable, and very distinct to those of 293 cells. To this end, we currently have a study ongoing where we have expressed proteins in the various XP-CHO protocols and then harvested at multiple days along the expression run, purified those proteins, and then we'll be performing analytics on them to better understand the critical quality attributes and how they change of these various proteins in the different protocols, so with and without temperature shift, as well as over the course of these relatively long expression runs that can be attained with XP-CHO. So as those data become available, they will be rolled out for review as well, and presented in the form of an application note. So the next question is, do you think you will continue to optimize the XP-CHO system to expand its applications and gradually phase out the XP293 system? So it's a great question. It gets a little bit back to my first answer, and I think the first answer is that the XP293 system will not be phased out in any way, shape, or form. In fact, based on the work that we've done through XP-CHO, we have identified significant areas of potential improvement for the XP293 system, utilizing some of the novel technologies that have gone into the XP-CHO system. So what we really see in the future is having multiple high-expressing transient systems that exist side by side and provide different functionalities for different proteins. And so to that end, we are looking in the future to be able to express a large variety of proteins in the different systems, and start to have, perhaps for the first time, if possible, and look at which types of proteins, which classes of proteins express better in CHO cells and which express better in 293 cells, so that you may eventually have multiple layers of decision-making process in terms of which system to start with. Again, whether you're looking for human or CHO post-translational modifications, and then hopefully at some time in the future also having a database to understand which is going to be your best shot on goal for that first expression system for a given class of protein. So I've had some questions come in with regard to the best practices for handling the XB-CHO-S cells, and whether there are differences, similarities compared to 293 cells. And as XB-CHO is a new system, there's a lot of learning that comes about in that system, and we certainly know that the system has been developed and optimized and pushed to an extent that we are able to generate very high titers of protein. But with that comes also the necessity to be sure that we're taking care of our cells as best as possible, because we are putting them into more stressful conditions. So at double the cell density of XB293, we are transfecting cells at a density really that has never been done before on any type of large-scale format. And we're asking those cells to produce protein for 12 or 14 days. So I'll answer this question in many different parts, and I'll just go back sort of to the beginning of when these cells arrive in your hands, and they're shipped from our sites to your laboratories. And some of these answers come based on actual experience working with many labs. So for a best practice for all cell lines as they arrive to your lab on dry ice is to really do one of two things. Either take those cells out of dry ice immediately, put them in liquid nitrogen storage for a few days so they can equilibrate, and then thaw them out as you normally would in a, for example, water bath until just a small amount of ice remains. Or secondly is to just thaw the cells immediately out of the dry ice storage. So allowing the cells to sit for extended periods of time on dry ice upon receipt, or the dry ice thawing and the temperature of the vials starting to rise and going through those transition points, or perhaps even worse so, temporary storage at minus 80 C. These are all practices that should be avoided. And when XB-CHO cells are thawed according to these best practices, we find that the cells thaw at a very high viability, and they begin to recover very quickly in culture. So it's important that these cells are thawed into 125 milliliter flask with about 30 mils of pre-warmed expression media, and the cells are immediately started on shakers. And as many of you are aware, in our protocols, we typically look at about 125 RPMs, plus or minus 5, depending on different shaking systems. But it's very important that the XB-CHO cells are constantly remain agitated so that they can have correct aeration for their growth. But what we do see is that the cells should take off in growth almost immediately, and by about 48 hours after thaw, the cells already start to get into their log phase growth pattern. And within about 1 to 2 passages, really the cells should have settled down into their 18-hour doubling time phase and should be very consistent at that point going forward. If for any reason the cells appear highly clumpy or even stringy out of thaw, there are certainly instances where either during transit or upon receipt, the cell vials have thawed, and then potentially been repacked in dry ice and refrozen, and that can certainly have detrimental effects. So I would say that if you're seeing anything other than nice single cells upon the initial thaw of the cells into cultures, stringy cells or highly clumpy cells, that is atypical, and those vials of cells should not be used. But the typical subculturing of the XB-CHO cells is fairly routine. As I mentioned in my presentation earlier, these cells will grow to over 20 million cells per mil in standard shake flask cultures. And it's important to note that their log phase growth does not start until about 4 million cells per mil. So it is important during routine subculturing. We try to subculture the cells only after they've reached 4 million cells per mil, and typically within the 4 to 6 million cells per mil window. But if you had to choose on either side of which was more detrimental, it's more detrimental to split the cells multiple times at lower densities when they have not reached their log phase growth. That can really stunt the cells, and sometimes they won't come back if they've been split at 1.5, 2 million cells multiple times. If they go over 6 million, much less problematic, 6, 7 million, no problem at all. If the cells overgrow considerably, 8 to 10 million, the best practice then is to simply split them back at a higher density. So instead of 0.15, 0.2, split them back to 0.5, should be no problem at all with the cells handling some time at the higher densities. We do recommend that a seed stock of cells is always maintained in the incubators, and this goes to a question that we had earlier today about passage stability, which is always a difficult question to answer because passage stability is based on many different factors, and many of those factors revolve around how well and how consistently have the cells been maintained during their routine subculturing. Meaning how often were the cells split before they should have, or perhaps overgrow significantly to very high densities, instances where shakers have stopped and aeration has slowed down. So all of these things play a role in determining what the passage stability of any cell line is, including XB CHO cells. In our experience, cells that are maintained well within that 4 to 6, 4 to 7 million cells per mil window will have excellent stability through 15 to 20 passages. There may be incremental losses in titer at those later passages, but again, that is somewhat dependent upon each thaw, how well it's been maintained and handled over that period of time. So when we talk about the cells as we start to scale them up for the transfection run, one of the things that we continually call out is the handling of the XB CHO cells. Again, very high density cells. And there is an excellent resource on our XB CHO homepage, or you can simply Google XB CHO YouTube. There's a YouTube video that was developed by the lead scientist on this project, Xiaoyan Liu. It's about 6 to 7 minutes long, and it actually shows every step of the way exactly how XB CHO has been performed by the scientists that actually developed the system. And so one thing that you'll take away from that video is how the cells are handled, and we've done some in-house testing on that as well, where these cells tend to be fairly non-clumpy during routine subculturing. You will notice as the cells get higher, 6, 7, 8 million or 8 million plus, that there will be some small amount of clumped cells in the cultures. We recommend not worrying about these cells at all. We recommend not trying to resuspend them by pipetting or by agitation. When you're splitting your cells or when you're utilizing your cells for the transfection, simply let those few clumps settle. Again, this should be relatively few clumps. If it's significant, that is atypical. But these few clumps can just settle, and then the cells can be pulled off of the supernatant suspension and used for all of those purposes. So no need to be concerned about them at all. And during the expression runs, the componentry of the system is developed in such a way that cell clumping will not be an issue during the expression run. So there's really no need to worry about any of the cell clumps. The other thing that you'll note from the videos is the way in which the cells are handled gently. So at all steps of handling the XB CHO cells, again, being a very high density, quick growing cell line, typically we recommend just gentle swirling at all steps. And in fact, we even did some studies early on where we looked at the robustness of the system between handling the cells with gentle swirling and with quite vigorous swirling of the flasks ahead of transfection. And that actually was one of the first instances where we were able to see that sometimes when the cells are handled very roughly ahead of the transfection, that you can see viability start to drop off in the expression run earlier than you would for cells that were just handled gently. So we always recommend that the cells just be gently swirled. They don't need any vigorous mixing to them, and that is the best way to handle those cells over time. So the cells at the time of transfection, so I had a question about what density should the cells be at the time of transfection. The cells should typically be at about 3 to 4, seeded to about 3 to 4 million cells per mil 1 day prior to the transfection. And that will allow the cells to attain approximately 7 to 10 million cells per mil at the time of transfection. So these cells now, of course, are at very high density, and with an 18-hour doubling time, you can imagine that actually your cell densities will change between the morning and the afternoon. And we recommend that once you have harvested the cells and grown them up to these high densities ready for transfection, that those cells are used for transfection and then discarded after that time so that they are not used again for routine subculturing. And again, in terms of passage stability questions, this is one area where you could have an impact if multiple times the very high density cells were continually used for repassaging cells for routine subculturing. So hopefully that's given a good overview on the XB-CHO-S cells. It is a very robust cell line. And just has some best practices associated with handling for optimal performance. So we had a question about the XB293 cells and whether or not we could potentially increase titers by transfecting those cells at 6 million cells per mil like the XB-CHO cells. And so this is another excellent question. So when the XB-CHO system was developed, we developed all of the componentry of that system to handle very high density transfection. And in fact, in certain situations where you may need to transfect even higher than 6 million cells per mil for very short expression runs, we have demonstrated that that is also possible within the componentry of the kit. So that entire kit has been developed to handle these very high densities and to handle them for very long periods of time. In contrast, the XB293 kit has been developed in a manner that is more amenable to 3 million cells per mil transfection for a 5 to 6 day runtime. And this is partly due to the different characteristics of the kinetics of protein expression between 293 and CHO cells. Excuse me, where 293 cells tend to have a big burst of protein expression early on, and then they relatively fizzle out, and the cell cultures will crash fairly quickly in the 6 to 7 day range. Whereas, as you've seen from the kinetics of XB-CHO cells, those cells take a little bit longer to ramp up their protein expression, but once they do, they will continue to express for a week longer than the 293 cells. So given the componentry of the XB293 system as it exists today, it does not support the high transfection density at 6 million cells per mil the way that the XB-CHO system does. And it would be unlikely in that case that XB293 would be able to perform in a similar manner as the XB-CHO system. Okay, so another question came in that I'm seeing cell viability less than 50% beyond day 7 post-transfection for XB-CHO. What can I do to improve that? So that's another excellent question, and something that we have fielded a few questions about in the past that my team has helped to troubleshoot. And the first answer to that question, it's certainly a multi-part question. This can have to do with scale, with the type of protein that you're using, and with the protocol. And I will say that from our experiences, we've seen that most of the time that we hear of decreased cell viability beyond day 7 post-transfection tends to be in the smaller scale vessels, such as the 125 ml flasks, and using the 2-feed max titer protocol that has a temperature shift associated with it. So what we've identified is a few areas that we feel are critical to improving that post-transfection viability, because the XB-CHO cells, even if they're sort of handled roughly, or some portions of the protocol are not performed as expected, they will typically hold in their viability for about 7 to 8 days before they start to drop off. But it is this high viability at the time of harvest that is the hallmark of the system performing optimally. And what we recommend is, firstly, going back to the first step of we're seeing that the first step is making sure that the cells are handled in the gentle manner that I spoke of previously. that tends to certainly lead to lower viabilities at later times in the expression run if the cells are shaken vigorously, et cetera, before transfection. The second is always making sure that the volume to flask ratio is what's called out in the protocol. So important that that is optimal or close to what's called out in the protocol, and that the RPMs of the shaker are matched to that volume and that particular flask. So in the case of the instances where we have heard of this viability dip, again, tending to be in the 125 ml flask size with the max titer protocol, we have a couple of different recommendations. Firstly, for this condition, the shake speed of the 125 ml flask can be increased. So if running at 125 RPM on a 19 millimeter orbital shaker like we have here, we have bumped up that RPM to about 140, and that seems to do quite a good job of handling that dip in viability post day 7. Secondly, we have also evaluated, only at the 125 ml size, the use of baffled flasks. And that has actually proven to be highly beneficial, especially for the 125 ml flask size and the max titer protocol. Now, the only difference in handling the baffled flasks in this protocol is that if you want to keep your shake speed constant, so for example, 125 RPM, the volume in the shake flask needs to be increased from about 30 mLs to closer to 35 or 40 mLs, where you have good shaking and gentle lapping of the media over the baffles in the flask. So what you really want to avoid is a lot of splashing, the media sort of slapping into the baffle, that you just have a very gentle mixing. If you wanted to keep the same transfection volume, so 25, 30 mLs, you would typically just slow your speed down a little bit from 125 to 115 RPMs. But for the 125 ml flasks, the max titer protocol, this has proven very beneficial to eliminating that dip in around day 7, day 8 viability of the cells. We have tested baffled flasks at other scales and found them to be unnecessary and or non-beneficial. It really seems that it's just this the small scale 125 where this dip is often observed, and that may be a workhorse volume for many of you. So certainly looking at handling the cells gently, speeding up the RPM, and or the use of baffled flasks is something that certainly can help troubleshoot that issue. And so along these lines, I've had some questions about the differences in the expecfectamine CHO transfection reagent. And this reagent, in terms of the best practices for using it, it is different than many transfection reagents that are out there, not in terms of, per se, the componentry. It's a lipid-based transfection reagent, but it tends to complex with DNA very rapidly. And many of you who have used the system realize that we call out to use cold OptiPro during the complexation reaction. And so we have done some significant studies recently helping to identify the most important keys to complexation. And those we have found to be twofold. One is the amount of time that the expecfectamine CHO spends diluted in the OptiPro. And we found that that time is best to be kept minimal, between 1 to 5 minutes. So dilute your expecfectamine CHO with your cold OptiPro at the time or close to when you're ready to add it to your diluted plasmid DNA. Hold times of longer than 5 minutes, 10 minutes plus for your diluted expecfectamine CHO will actually diminish the performance of the complexation reaction. So once diluted with the OptiPro, add that to your diluted plasmid DNA. And typically, we recommend to mix this with just gentle inversion or maybe 1 to 2 pipetting steps. We have data that you can go back and look in my slides where vigorous pipetting or vortexing can significantly decrease the titers achieved of those proteins. So simply dilute the expecfectamine CHO with your cold OptiPro, mix it with your DNA gently, and then hold that for anywhere between 0 hold time and about 5 minutes for optimal results. So with expecfectamine CHO, the complexation reaction happens almost instantaneously. So the moment that you have mixed your DNA and expecfectamine CHO together, they're ready to go into the flasks, and we recommend a hold time of no more than 5 minutes. Now, we do get questions on whether or not a direct transfection can be performed, whereas the plasmid DNA is added directly to the flask and the transfection reagent is added directly to the flask. With this particular reagent, we have not seen success in that. It needs some period of time to complex with the DNA, but again, a very minimal period of time. Now, in instances where a short hold time of your diluted transfection reagent is not desirable or possible, we have, for example, in our application note for the scale down protocols using 96 well deep well plates, 24 deep well plates, or also the 50 mil mini bioreactor tubes, we utilize a procedure where the plasmid DNA is actually diluted in the entire volume of OptiPro that would normally be used to dilute the transfection reagent and the plasmid DNA. So it's in about double volume of OptiPro. And then the non-diluted expecfectamine CHO transfection reagent is added directly to that diluted plasmid DNA. And for small-scale studies, that makes it much easier, but that may also be, if you are concerned about the amount of hold time that your transfection reagent is going to see in the OptiPro, we recommend just doubly diluting the DNA and then adding in your non-diluted transfection reagent. And along these lines, I've got another question on the plasmid DNA about the optimal amount of plasmid DNA to be used. And this one is quite interesting because 1 microgram per mil of transfection volume has really been a paradigm for transient transfection for quite some time. And we've actually found that with XP-CHO, the transfection reagent is so efficient that really using any more than 0.7, 0.8 micrograms per mil of DNA has been unnecessary in our hands. And in fact, the sweet spot has been closer to 0.5, 0.6 microgram per mil of DNA. And so we can actually see in instances where significantly more DNA is used, a few percentage points of drop in viability of the cells over time, simply because the more DNA that you add, the more that you transfect in, the more stressful that transfection is to the cells. And we're already utilizing a very highly efficient transfection mechanism that highly transfects the cells and certainly does impart some stress on the cells. And so from a standpoint of both saving time and money in your plasmid DNA preparations, as well as utilizing the high efficiency transfection of the system, we certainly recommend 0.6, 0.7 micrograms per mil, probably as a better place to be functioning with regard to DNA concentrations. And in the case of monoclonal antibodies with a heavy and light chain, that would constitute the total amount of heavy and light chain, so a final overall amount of DNA at about 0.6 to 0.7 micrograms per mil. And so I see a question about the addition of the XP-CHO enhancer and feed to the cultures. When is the optimal time to add the XP-CHO feed and enhancer? So this, as called out in the protocol, the optimal time that we found has been 22 to 24 hours post-transfection. So this allows the cells to be transfected, to go back into the 37C incubator for an overnight period to recover, and then on that next day have the enhancer and the feed added to them. And one thing, again, that I mentioned earlier in my presentation today is that neither the feed nor the enhancer needs to be pre-warmed. And in fact, one of the easiest best practices to do is to pre-mix the feed and enhancer before you add them to your cultures. Certainly at small scales, that's quite beneficial so that you're not having to micropipette 96 well plates with 2 different solutions. And even at larger scales, that reduces the number of additions that you have to do to each one of your flasks. So again, these may be mixed ahead of time. We have demonstrated up to 2 weeks of stability for holding these, but I would suggest as a best practice to dilute them fresh ahead of time, ahead of use, and then add a single addition to the flasks. So we got a question about the successful examples for expression of intracellular proteins and how was XB293 compared to HEC293, and then I'll also mention XB-CHO here. So we certainly have generated, for XB293, generated some data in-house as well as significant external data on the expression of intracellular proteins. And the results have, and feedback has been overwhelmingly positive in terms of expressing intracellular proteins in XB293, as well as the boost that you get compared to traditional 293 systems. Certainly here as well, the intracellular proteins takes advantage of the greater biomass of both of the XB systems, in case of 3 million cells per 293 and 6 million cells for XB-CHO. But intracellular proteins have many interesting caveats of their own, including how long the expression run goes, the potential for toxicity to the cells. And so we are in the process right now of working to finalize some data of a panel of intracellular proteins that was previously expressed in XB293 that expressed at either high, mid, low, or no expression levels, and comparing those to the same proteins expressed in XB-CHO by the 37C or 32C protocols. And so while we don't have a full data set there yet, what we certainly do know from some upfront data is that it's important to understand that intracellular proteins are very different than secreted proteins, as I'm sure you're all aware, and thus the takedown time and the lysis of the cells is important in terms of both proteolytic degradation and viability of the cells. So oftentimes we hear many users in XB293 expressing their intracellular proteins for approximately 3 to 4 days. And so we hope also that in the case of the XB-CHO system, that a 3 to 4 day expression run, taking into account the higher biomass of the system, and even potentially tweaking the system for in ultra-high density transfection, will also allow for excellent expression of intracellular proteins. From data that I have gathered externally, I have heard that XB-CHO does a nice job of expressing intracellular proteins, some slightly better than XB293, some slightly lesser, and some quite comparable to XB293. But hold tight on those, and we hope to have more data in the near future, a direct comparison between the systems. Then we had a question about making stable cell lines from XB293 or XB-CHO cells. And will they express higher than normal 293 or CHO? Here we have certainly direct knowledge of the CHO system. We had previously taken XB-CHO-S cells and performed stable cell line or stable pool generation compared to very well-defined systems for doing so in our GMP CHO-S cells that are routinely turned into stables at our Gibco site. And what was very interesting is that this pattern of high expression with the XB-CHO-S cells certainly did translate into stable cell lines. And it was the initial work that was done. It has not been exhaustive at this time. But it was a fairly rudimentary optimization of the culture conditions for the XB-CHO-S cells because they do grow differently than the CHO-S cells. But what we saw is that within the stable pool, we were able to hit gram per liter levels with the XB-CHO-S cells, and some of those levels being multifold higher than what we would see with the parental CHO-S cells. So the one caveat here, however, is that when we looked at the profiles of the XB-CHO-S cells, we noticed that oftentimes the viability and the density was beginning to drop simply because those cultures and the conditions had not been fully optimized. So we expect when fully optimized in a head-to-head comparison that in fact the XB-CHO-S cells will make a very good stably expressing cell line. Then we got a question about the best time to harvest supernatant, and I think that flows nicely into the question about the intracellular proteins as well. So we have developed the XBCHO system, especially in the case of the max titer system, to be able to have high viability under optimized conditions well out to day 12, 14. And in our hands and in many end users' hands, they have taken these cultures out with very high viability for 19 or 20 days or even more. Now, with that being said, while the cells will maintain high viability, they do stop producing protein. Of course, in cultures that are maintaining somewhere between 8 to 10 million cells per mil during the expression run, you're certainly going to have a buildup of metabolic waste over time, lactate, ammonia, and many of these things will trigger the slowdown or stoppage of protein expression. So we typically recommend that for the standard 37C protocol, day 7, day 8 is a pretty good takedown time. Now again, data was shown earlier in the virtual event today of taking proteins out to 10 or 14 days at 37C. That certainly may be beneficial for some proteins. Also in one of those examples, there was a decrease in titer between day 10 and 14, so there could have potentially been proteolytic degradation at that time. So in general, we recommend about day 7, 8 for the 37C standard protocol. Somewhere between 10 to 12 days maximum for the high titer protocol, and probably 12 days maximum for the max titer protocol. So while that will go out to 14 days, you do hit a point of diminishing return, and of course you're going to then have the propensity for proteolytic degradation, lysed cell debris in the cultures, etc. So it is recommended, as always, to harvest when cell viability is high, and again in that 7 to 8, 8 to 10, 10 to 12 day range for the various protocols. Then I had another question about sometimes the supernatant for the XBCHO system is difficult to filter during clarification. This is, again, a great question and something that we are certainly aware of and are addressing currently. Lena and Panda on my team earlier presented on an optimized clarification and protein A purification protocol for the XBCHO system. And in terms of clarification of the supernatant, there are certainly differences in the componentry of the XBCHO system for CHO cells and the XB293 system for 293 cells. So I do think it's logical to not expect that the two will port exactly into purification, clarification protocols from the other system. And so what we have seen generally works best for XBCHO is a centrifugation period to clarify the supernatant of cell debris, and then initially passing the supernatant through a 0.45 micron filter, and then through a 0.2 micron filter. And this allows for relatively easy clarification through the basic bottle top vacuum filters, which really represents sort of the lowest end in performance of filtration. So other filters, depth filters included, handle the XBCHO supernatant without any problems at all. But it is at this sort of more basic filter top, bottle top filtration, where there can be some clogging of those filters. And again, I would recommend, instead of trying to go through multiple 0.22 filters, do the pre-filtration in 0.45 micron, and then the 0.2 micron filtration should go much easier. Now, that being said, part of the presentation earlier today, Lena pointed out that we had another customer inquiry about a yellowish tinge to some proteins that were purified using protein A. And we have done fairly exhaustive evaluations to understand what the componentry is in XBCHO that differs from XB293 and leads to some of these, some of this yellow discoloration of protein A purified molecules. And we have, in fact, identified what that component is, and are currently working to validate the reduction in the concentration of that component, which in our hands appears to be fully seamless in the transition. So obviously CHO medias and feeds and componentry is tailored a bit more towards the bioproduction versus the research scale end of things. And bioproduction processing and purification methodologies and equipment are extremely robust to handle these types of things, whereas typical bottle top filtration is less efficient along those lines. But I do want to make it aware that we both have heard these concerns, have identified the issue, the root cause, and are working now to implement a fix, which again should be absolutely transparent in terms of the performance of the system. To that end, I will reiterate what Lena presented earlier today with regard to optimizing the Protein A purification protocol. And what Lena was able to find, and I'll just briefly summarize this as you'll be able to look back to her presentation, and then there'll be an application note coming out shortly as well, is that the vendor recommended conditions for many Protein A resins utilize phosphate buffers for the loading and elution buffers. And what we found what Lena has found, is that simply changing from phosphate buffers to tris buffers allows this contaminant, this impurity, to be washed from the columns, and the protein that is bound to the protein A column, when eluted, the eluate has no discoloration. The column has no discoloration. It is completely removed, and even in the system as it exists today, this simple fix that, again, you can find in the presentation or contact us as necessary, that will be in an application note shortly, simply switching to these tris buffers completely eliminates that issue. But because this component is also important in the filtration aspect, we were glad to see in one sense that it is the culprit for both the filtration difficulty as well as the protein A discoloration, so that when we successfully look into modifying the concentration of this component, we should be able to address both of these simultaneously, so that even in the absence of the workarounds that we have provided, it should be relatively straightforward in terms of handling the supernatant and protein A purification more consistent with existing methodologies. So if there are any other questions, I'll be happy to field them now. I think these were excellent questions. Many of them are covered in the frequently asked questions that I have incorporated some of the upfront questions that we received and have had discussions with end users around into the end of my slide deck. And they are there for your reference. And we continue to look forward to sharing any tips for optimizing the system, as well as for optimizing any problems that arise in your work. And really appreciate all the feedback and the collaboration that we have had with our users of the XB systems for many years now, and hope to continue to provide the level of support that is required to help you achieve all of your goals with your transient expression systems. So thank you very much for joining, and we look forward to continuing to collaborate with you all in the future.
So since there are no more questions, I would like to once again thank Jon for his expert opinion on mammalian transient expression. I would also like to thank Gibco, part of Thermo Fisher Scientific, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through May of 2017. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.